Start. I saw we're good to go.
Sure.
Okay, wonderful. Well, thank you all so much for joining us today for our monthly George M. O'Brien Kidney Resource Alliance, or OKRA, National Forum. Today, we will be hearing about some technologies available from the Northwestern National Resource Center, and really happy to kick this off. Just wanted to remind everyone and give you a couple of quick announcements. If any of your trainees are sending in abstracts and getting them accepted for spring meetings that are kidney related, the OKRA consortium does have some small travel awards. We reimburse up to $500 for trainees who are the first and presenting author on an accepted abstract. There's more information on our website, so we can maybe put that link in the chat. You can navigate there, and it's on our homepage as one of the sliders, but that process is open. And so if you've already gotten an acceptance, or if you have someone working with you who might be eligible, please feel free to share that information with them. That, again, we have open for the next month or two before we need to close that down for spring meetings. We also just wanted to announce that on the main website, there are recordings from other interactions and other webinars that we've had as a whole consortium. So please feel free to check out the website, see what's going on, see what opportunities are available there. But also we have several links to other presentations that might be of interest to you that highlight resources that have been shown over the last couple of months. So thank you for joining us again. Am I missing any announcements, Demetria or Mary, from the OKRA Coordinating Center end?
Nope, I think that's everything.
Great, thanks so much. I'm happy to turn it over to Sue Kagan, the PI from the Northwestern O'Brien National Resource Center. Thanks for hosting this today.
Okay, well, thank you very much, Amanda, and we're thrilled to be here on behalf of the Northwestern and USC O'Brien Center using physical sciences to advance kidney health. And I'm thrilled to introduce today's presenter, Dr. Ranjit Singh. Randy is a Moderna Global Fellow and Assistant Professor here at Northwestern University, working in the Kelly Labs in the Department of Biochemistry and Molecular Genetics. Randy is a fellow Canadian, so I'm particularly thrilled. He received his PhD at McMaster University in Ontario before moving to Boston to do his postdoctoral fellowship at the Mass General and Harvard, where he stayed on as faculty before being recruited back to Ontario, now to the University of Toronto, by Dr. Shana Kelly. And he was there for a couple of years before moving with the Kelly's lab here to Northwestern University. And his areas of focus and what he's going to talk about today is something that we're really excited about. Large-scale phenotypic screening technologies that enable us to do all sorts of things for all sorts of diseases. But with respect to kidney diseases in particular, thinking about identification or isolation of rare or difficult to isolate cell populations, identifying drug targets for classically undruggable targets, as well as uncovering pathomechanisms. So I'm going to hand it over now to Randy and look forward to a great session. Thanks.
Thank you, Sue, for the kind introduction. Hopefully everybody can hear me. Can you just give me a thumbs up? Since I'm used to presenting this technology across people from very different backgrounds, I always start with a disclaimer, which is that, let's see here. Just a quick disclaimer. I'm not a nephrologist. Since this is an audience of nephrologists, I want to make sure before you sharpen your knives later on in the talk, just be aware that I was lucky enough to be standing next to one of the best ones in the world. So, I have allies who are really good nephrologists. If I need to, I may not be a nephrologist. Now, with that being out of the housekeeping that we're taking care of, I wanted to start by asking a very simple yet powerful question to the audience that has gathered here today, which is, what groundbreaking advancements in kidney disease research could you achieve if you had the power to sort over 1 billion cells in under an hour? Now, I'm super excited to share with you today that that level of remarkable throughput is a hallmark of a platform technology that we in the lab refers to as LeaPFroG. So what is LeaPFroG? Now, LeaPFroG stands for Large-Scale Phenotypic Functional Regulators of Genomics. I know it's a bit wordy, so we can try to look under the hood, try to figure out what exactly does that mean, right? So since we treat LeaPFroG as a discovery engine, I'm going to use that analogy to sort of describe what it really means. So let's look at under the hood, if you will. So I'll break down each component of those letters to see what do they actually refer to and how they actually fit in together. So large scale comes from the fact that we are utilizing microfluidics-based devices to be able to sort cells at scale. Now, that capability is made possible through two components. One of them is the approach itself, which is that as opposed to, say, a fluorescence-based cell sorting, we utilize magnetic ranking cytometry. So in comparison to FACS, we actually use immunomagnetic nanoparticles to be able to detect a antigen of interest, and then the level of that particular antigen at each cell level serves as the basis for sorting cells across a population to be able to stratify based on the level of expression of that particular antigen. Now, that is made possible through these microfluidic devices. Now, we have a couple of different flavors of these devices. We typically refer to them either in a prism format or a cytosort. And the key things for you to remember about these two sort of flavors is that they're just a matter of how we actually sort the cells in terms of where they end up, right? So in the case of prism, that architecture is specifically designed so that as the cells are going through the device, we actually pull out the cells that we want of importance. Right? So that way, majority of the signal will actually, majority of the cell population actually will actually end up in the low or no expression bucket, because we're really only interested in rare cells. Now, in applications where we actually are interested in large population cells that do have the signal, we typically utilize what's called a cytosort architecture. And in this case, we're not actually pulling any cells. We're in fact enriching the cells within the device itself. Those two characteristics of these microfluidic chips is what allows us to be able to utilize these devices across whole plethora of applications, as you'll see as we develop through this seminar. Now, under each of these two architectures, we actually have a whole series of additional devices. So for example, under the Prism, we've devices called NU Prism. That actually is specifically designed to be able to sort isolated nuclei in applications where we're only interested in, say, the differences in expression levels at the nuclear, inside of the nucleus, while not paying attention to what's going on at the apical surface or other areas of the cell subcellular localization. That chip is pretty powerful to be able to do that analysis. And similarly, ExoPrism is specifically designed to be able to sort exosomes so that we can actually just utilize that capability for analysis of enriching of exosomes. And under the cytosort, we have a whole family of devices. The one that I want to highlight, two that I want to highlight actually are what's called a ProSlex, which is a device that we've designed to be able to do directed evolution-based enrichment of novel aptamers. And the second one is, which is going to be relevant to this audience, is that I'm introducing a new device, and I'll walk through how that device works. What we're referring to as, for now, as NephroSort. So how does LeaPFroG enables large scale? And that has to do with the fact that we can easily parallelize. So let me give you an example. So for example, individual chip, here I'm just highlighting, for example, the size of this device. As you can see, the Sharpie is placed next to the chip on the right. And that's about the size of this device. And it can actually sort 20 million cells per hour. But we can easily do sort of like a daisy chain linking of these devices to be able to expand the capabilities. So as you can see, the footprint itself did not increase, but we've actually increased the throughput. So now we've got 100 million cells within the same sort of small region of space that we occupied. And if you're really ambitious, you want to be able to say how far we can push it. We've actually demonstrated in a seminal publication looking at the CD47 regulators that we could actually do this kind of daisy chain linking to be able to demonstrate capabilities up to 1 billion cells per hour. So that explains the large scale. Now, the phenotypic part of the LeaPFroG refers to the particular features that you would be interested in, right? So these could be, for example, cell surface expression differences. So maybe there's a particular protein that you're interested in seeing what's the level of that protein on an individual cells. Or it could be even a modification, right? Say a state of post-translation modification, say a phosphorylation status for a particular protein of interest. We're not restricted to the cell surface. We can, in fact, do intercellular proteins detections as well. And as you'll notice on the slide at the bottom, I've highlighted how the phenotypic readout complexity is actually emerges as you go to a specific type of applications where we're more and more sort of confident in how we can deploy the technology to be able to address harder and harder questions. So for example, I mentioned cell proliferation here. It's quite complex because now you have to deal with not only just the fact the presence of absence of a signal, but also the cell state, underlying cell state that is actually contributing to that particular level of cell signal. And the last one is secretion dynamics. This is probably one of the most complex because it's no longer about individual cells, but their interaction with their neighbors, right? So up to the point where we actually published the first seminal publication last year describing the use of this type of technology for understanding cellular secretion dynamics, often the field has to rely on actually blocking the secretion, right? So you would actually just use some sort of a cellular intracellular trafficking blocker so that in fact no secretion occurs. What you're really measuring is the accumulation of that particular signal inside the cell. Now, if you really want to study secretion, it's not just the secretion, right? It's the autocrine and paracrine interaction of those cells that are actually influencing those secretion rates. So that accumulation within the cell doesn't really capture the right sort of environment where those changes are taking place. So this should highlight to you how defining a particular assay that actually fits with the right phenotype of interest or the question that we want to address is key to be able to actually gain significant advances in making progress towards novel discoveries. And so the last part, which is the functional regulators part, right? Which is that refers to as how we actually going to utilize the whole unbiased approach to be able to address what are the potential regulators of these particular phenotypes of interest, right? And that comes from utilizing collection of genetic perturbation libraries. Now, these libraries can come at different sizes and scales. They could be genome scale, or they can be different types of libraries. So for example, in the case of CRISPR, it could be CRISPR initiators, activators, inhibitors, or activators. And there's a whole different sets of libraries that one can utilize. And there's also CRISPR knockout libraries. The most common that the field has used is probably Gecko and TKOV3. Those are the ones that we utilize in the lab. And then there also is the custom build libraries. These are the libraries that you built specifically for a very specific question that you've asked, right? Where you're not necessarily concerned about the whole genome. You're just concerned about a subset of the genes that you know are likely playing a role in a particular phenotype of interest. And now it comes with a sort of a drawback, right? So as you can see, in this case, the genome coverage or the diversity will actually go down as you go from left to right, where if you do a whole genome unbiased, of course, there's a high diversity, but when you go to the custom ones, there's going to be low diversity, but it's very high specificity. And then the last one is where the magic happens, right? The genomics part. Genomics part refers to how we actually layer all these different components that LeaPFroG is built on to be able to demonstrate how we can actually address very, very challenging questions. And we've actually done that across multiple different projects in the lab and with partnerships with other groups. I want to highlight a few of them that are actively actually taking place within our Simpson Cray Institute labs. These ones involve, for example, the identification of checkpoint inhibitors. This is the CD47 story that I just mentioned briefly. And also in vivo screening for drivers of tumor progression, which we can actually try to understand what actually drives these cells to metastasize. Can we capture those, for example, circulating tumor cells that actually has left the original tumor site and actually invaded different areas of the body, and which we know are to be the most harmful to the patient. And also, as I mentioned, because we've been able to demonstrate that we can do the complex phenotype analysis at single cell resolution, we've done proof of concept experiments with T cells, for example, using IBD and GVHD as a sort of a proof of concept model to show that we can identify genetic regulators of interferon gamma, for example. And there's, as you can imagine, there's many other applications you can build in. So we've actually even, have set our sight on even harder and harder problems as we've progressed, as the platform has matured. So the latest one, for example, is trying to be able to do progress on finding actual genetic modulators of previously undruggable targets. And there's an active project in the lab that's going on that targets, for example, KRAS, which we know to be one of the top oncogenic genes in oncology. But for the last 30, 40 years, even though we know a lot about the biology of the protein, there's very little sort of progress has been made to be able to make actually tractable, durable treatments in the clinic targeting KRAS. So, as you can imagine, this type of capabilities requires an architecture that is, even though we're not, sort of officially a company, we pride ourselves on organizing in a manner that actually operates like a company, right? We have a visionary leader on top, Professor Shana Kelly, who sort of sets the sort of basically the standard for how we're going to operate, right? And then you have the next set of people that are operating with that vision in mind, and that includes myself and two of my very, very close compatriots, Daniel Wang and Kang Fu Chen, who really are the brains behind how the actual technology works, right? These guys are so good at it that you tell them, hey, how can we tinker this to this particular application? Within a week, they'll come with a format that's going to be, oh, okay, let's give it a try, right? So our job is to be able to make the right tools available for the particular problem that has been identified by us or other collaborators. And then you have the first generation of LeaPFroggers, right? These are the believers. These are the people that say, hey, okay, there's a specific problem that I want to address, and I think this technology can help me. And they've actually demonstrated, each one of those people that are listed on this slide have actually demonstrated with a specific project that they can actually make progress on that particular problem by just utilizing this technology. And given the success of these, and original OGs, if you will, now there's a whole next generation of actually LeaPFroggers, which is a bigger generation than the original ones. And these guys are now actually taking that technology even further, once again, pushing boundaries in terms of what kind of question we're asking with this technology. And my hope is that after this talk, I'll be able to at least inspire a few of you that there's a room for more people to be able to utilize this technology for the problems that are close and dear to your heart. So, but before we get to that, I do want to point out, there's a sort of code that we follow as LeaPFroggers. And I'm solely this line from one of my favorite book, which is the Hitchhiker's Guide to the Galaxy. So we call it the LeaPFrogger's Guide to the Galaxy. And there's the three basic tenets of this sort of philosophy, which is that you have to pick hard problems to solve. If you're going to wake up every morning, you want to work your 24 hours, whatever time you can actually dedicate to that specific problem, try to solve a harder problem, because that's going to make a bigger impact into the world. And the second is, if you're going to pick a harder problem, you better come with a can-do attitude, because you're guaranteed a failure across many different steps of that, when it comes to hard problems. So you got to have the attitude that, a single failure or a single setback is not going to stop you from solving the problem. And the third is that prepared to put in the work, So that just basically goes with anytime you want to solve hard problems, it's going to come at the cost of you have to put in the hard work, right? So when we preach, as a pep talk to any of the new LeaPFroggers, one of the things we always say is that, I'm willing to compromise on the first one, right? Maybe for the first little while, I'll let you play with this, maybe perhaps a smaller, not as hard of a problem, just because you want to be able to teach you to be able to walk before you can run. But when it comes to number 2 and 3, there's no compromise or replaceability. So as a LeaPFrogger, you better embody those 2 and 3 points that are highlighted on this slide. And as I mentioned, this technology didn't just emerge today. We've actually working actively on it for the last 7 years, right? Starting with the very first description of the PRISM architecture paper that details the physics and the microfluidics and the whole shebang about describing in detail how the platform actually works and how effective it is compared to the existing technologies. And then ever since then, across the last, as you can see, as we've looked through this evolution from that point on, we've started to deploy this platform across harder and harder problems with success at each point. Now, there were many failures along the way. That only actually inspired us to actually design different devices to be able to go back and say, look at the data. What did we miss the first time, right? What was about that specific problem that the previous approach that had worked so well didn't work for this particular problem? And that actually resulted in a generation of those additional devices. For example, the NU PRISM emerged because, as Abdullah was trying to sort what are the genetic regulators of beta-catenin, he found that our basic PRISM device doesn't work because beta-catenin has 2 pools of population, right? There's the apical pool that sits on the plasma membrane, and then there's a nuclear pool. It's the nuclear pool that is critical to be able to understand the regulators of that pool. But when you deploy PRISM, it cannot tell the difference between the apical pool and the PRISM pool. So for that, we have to design an NU PRISM, which can tell the difference between a nuclear pool versus the apical pool. And as I mentioned, we've been sort of getting more and more ambitious in the type of problems we're pursuing, to the point where we're right now actually utilizing these technologies to be able to address one of the hardest problems in oncology, which is cracking KRAS. And we've made considerable progress, and that paper is currently under reviews, and I'm hoping to be able to share that at some future time. So the question up to this point, my sort of hope is that this has made it clear to you guys that this is not a technology that was just coming up, right? We've been at this for the last 6 years. Now the question is, how can we deploy this technology for specific problems in nephrology? And we were fortunate enough that when we initially published the CD47 paper, after we sort of celebrated the publication and describing that we were the first one to figure out what are the genetic regulators of CD47, after sort of having a little bit of a champagne, Jenna comes and says, okay, Randy, what do you know about IgN? And, I didn't want to say I don't know anything. I said, I can. It's close enough to IgN, right? Well, she says, okay, well, here's a Dropbox link with some IgN papers. Why don't you go and see, what you can come up by Monday? Because if you could, we should probably apply for a Genome Canada grant. So I took a look at those papers, and I'm so grateful that in those papers that she has selected, she had kindly included a primer. These are one of my favorite set of papers, which is I don't know the field, but these primers give you enough so you actually get an idea of what is the field all about, what are the sort of key problems, and how you can potentially think about addressing that key problem, right? So it was clear from those IgAN primer papers that the pathology was very unique, right? So in IgAN, you've got this observation that you've got these deposits in the kidney, but they're not just random deposits. It turns out they're specific to a very certain type of cell type, mesangial cells. And that was, as a cell biologist, that was kind of, sort of going around, sort of, what do you call it? A eureka moment, if you will, say, why only mesangial cells, right? So why would only the mesangial cells are where these deposits are taking place? And then when you were thinking about what is the actual current best understanding about what's actually driving the development of IgN, you come to what's called a multi-hit hypothesis, where it's been sort of known very well in the field that there are multiple hits that take place for that final phenotype to emerge, starting with that very first one, which is that for some reason there are increased levels of modified versions of IgA1, particularly the galactose deficient version of the IgA1. And because these are foreign to the body will actually generate autoantibodies to these galactose deficient IgA1. And then these autoantibodies, then as the antibodies are making to the circulation, they actually form complexes called circulating immune complexes with additional proteins and molecules, including IgM and components of the complement pathway. And then this circulating immune complex is what ends up actually deposited on these primary mesangial cells. So with this information in mind, I thought, okay, this is a pretty easy problem. I can probably crack it. So I was like, okay, here's what we should do. So I mentioned this to Shane. I say, well, we'll just propose identification of cellular determinants impacting mesangial cell specificity in IgA. We'll partner with a receptor. Chinook Therapeutics was more than excited to be that receptor, because that was one of the requirements from Genome Canada, saying you must have a company that is willing to believe in what you're doing, because they're going to be able to take the stuff that you've generated and then potentially have a path towards translation. And I thought, hey, this sketch was pretty cool. It's crystal clear to me, right? I'm going to have a primary mesangial cells, I'm going to have a non-mesangial cells, and I'm going to utilize some sort of specific library because I figured if it's the mesangial cells, it's probably something on the cell surface, right? Because how else would you be able to tell that there's a difference between mesangial cells and some other neighboring cells, right? So to me, it made sense to just start with a surface cell library. And then I was like, I'll use different types of sensitizers, perhaps a galactose deficient IgA, and in combination with more complex types of these sensitizers, including the IgN complexes, if you compare from the people, or have collaborations from experts in the field who have been able to engineer these circulating immune complexes. And then I just want to be able to then collect large amounts of data, and that's the throughput that I had in mind, right? Is that for this kind of a capability where I want to do multiple parallel analyses, I want to be able to do them at scale, right? So that I can actually have that giant, data set that will allow you to be able to compare. Okay, what was the difference between the same sort of treatment if the cell line is different, or if the genetic insult is different? So I thought this is a no-brainer. It should get through funding, no problem. Of course, Genome Canada had other ideas. They came back basically to what I think were them sort of main feedback. They said, neither Professor Kelly nor Dr. Singh or Atwal, neither one of these investigators is a nephrologist. And the second feedback they gave us was, well, IgN deposits most likely just bind transferrin receptor, because we know that transferrin receptor shows up on genome-wide analysis studies. So what else could it be? There's nothing new here. There's no way, this is actually worth funding. I was like, well, this is true. Let's take a moment to reflect. What are they really trying to tell us? Well, it's true, I'm not a nephrologist, so that critique was valid. But then this other one, saying transferrin receptor argument, I was like, well, there has to be data that we can look up to see if does it make sense. So luckily for me, we're living in the sort of age where lookup tables are available, right? The field has made available tremendous amounts of data, which we can utilize to address that question. Is it really the transferrin receptor? So here, for example, what I'm showing is a single cell RNA-seq data that's been collected as part of the Atlas KMP organization supported by, again, NIDDK. where you can isolate specific different cell types from the kidney and then ask the question, what are the expression patterns of transferrin receptor? And what I want you to highlight is the data on the right, which is the actual expression patterns of that transferrin receptor. So you can see, if this is the area where specifically the MC refers to the mesangial cells, right? If those are the mesangial cells, I can't tell you that there's any noticeable difference in expression between those cells or any of the neighboring cells or any of the neighboring clusters, right? So clearly, this to me indicated that we were thinking of the right way. It's not the transferrin receptor that is likely contributing this deposit. So there has to be more to this, right? We were thinking, hey, NU happens to have one of the best nephrologists in the world, and Shana Kelly, being a chemist, knows the importance of a catalyst, right? So if you, for a right kind of a reaction to occur, you need a right catalyst, and there could not have been a better catalyst than Professor Quaggan, who immediately saw the possibilities, right? Which is, she's saying, okay, wait, this actually makes a lot of sense. Why don't we try it, test it out? And this will be great to include in our U54 grant. And we were fortunate enough that we identified as a small component of the U54, we sort of changed the scope a little bit. We said, let's put a little more words so it becomes a little more clear, right? So identification of cellular determinants impacting selective vulnerability of mesangial cells in IgA nephropathy. So it's not really a simpler one. There's a little bit more bigger words. And surprisingly, it got actually funded. So we were so grateful that we were able to actually have a particularly well-defined deliverables and activity that will allow us to be able to pursue this original idea that he proposed to Genome Canada. So, as they say, if you build it, he will come. That's one of my favorite movies, a quote from one of my favorite movies, Field of Dreams. So that's what we wanted to do, right? Okay, let's go build it. So what is it going to take? So first thing we had to do was develop a cell surface library. We had looked into the literature. There wasn't one readily available. People had generated cell surface libraries, but they were all sort of proprietary, right? So we're like, okay, it's not a hard problem. We know what cell surface library should include. We've done a basic sort of search on key components that would be included. That was through partnership with, for example, even just looking at commercially available ones. It turns out, you can buy from Thermo, there's a 750 gene. So at least I got that 750 gene list, right? But then we did additional analysis because 750 gene wasn't encompassing all of them. We did additional in silico analysis to be able to identify all possible cell surface disruptors across all possible cell types. And that resulted in about a cell surface library of about 3,000 cell surface proteins. And we wanted to be able to show that we can have multiple guides targeting each one of those genes. So we built a library that was in total about roughly 12,500 elements, and where we included enough controls to ensure that we can actually see the differences between a on-target versus an off-target effects. And that was sort of prepared using the standard semiconductor electrochemical-based synthesis. It was in collaboration with Zinstrict. And I'm showing at the bottom on the right, the library actually is pretty good. All the statistics would indicate that this was ready to go into the cells. Now, before you can get it ready to the cells, though, you need something else. You need, again, to be able to have the sensitizer, right? This is where, the whole quote about, if you build it, he will come. I was so fortunate and lucky to be able to have a student actually, who had recently graduated and was just doing, preparation for MCATs before he starts med school. And he was looking to do something more productive with his time. He's like, hey, this looks like a challenging problem. I want to have to take a crack at it. And he says, well, okay, I know that IgA glycosylated pathway can be defined in a manner where we can have a particular area where we can make a mutation to be able to bias that towards galactose-deficient IgA production. So that's a very simple problem. We'll just use a knockout to generate, for example, take Dikiki cells, B cells that produce antibodies. We'll just make a knockout. So now those Dikiki cells are producing mostly galactose-deficient IgA. So we'll generate that, and that becomes our sensitizer. And then since we already have a library, all we have to now do is combine the two, and that should be doable pretty easily. So that's in fact what we did, right? So it's very simple. We took those cell surface library, we took the primary mesangial cells, and we used that galactose-deficient IgA as an initial sensitizer to be able to say, can we find differences in population of cells where it's the galactose-deficient binding itself that will be the key feature that we go after, right? Because if those deposits are happening on the primary mesangial cells, we know they have to have included galactose-deficient IgA as the component. So we did the enrichment of low versus high populations, and we did the full data analysis that we typically do. And for that, I'm grateful for Chen Lin Zhao, who's a PhD student, who among her other projects just said, hey, looks like a pretty good project I want to help a lending hand to. And she did the help with the analysis of the data. And so we have a few hits identified, and we wanted to be able to use orthogonal assays to be able to see, are we actually picking a real signal here, right? So I'm not identifying the gene here, but I wanted to show you that in the quadrant where we would think, significantly enriched genes should show up with a high enough statistical power to ensure that they were not false positive, that we can actually look at, existing data sets where we can say, okay, there exists the IgA Gloom data set, for example. Is the expression of this particular gene different in the healthy versus the IgA nephropathy patients? And to our surprise, and actually confirming what we believed was going to be the right approach anyways, we do in fact see that there were great differences, statistically significant differences between when you compare the expression of this particular gene in healthy individuals versus the IgA nephropathy patients. So that's a one line of evidence that you have. It's always great to have multiple line of evidence. And for the multiple line of evidence, we went to the in silico data mining, looking at cell type specific expression differences. Again, we're leveraging the stuff that's already been built by the field, right? This is the single cell sequencing data where we can say, okay, I know which particular gene I'm interested in. What does the expression pattern of this gene looks like across the population of kidney cells, right? And what I was really amazed to actually discover and find was that the genes that we identified, in fact, had that critical attribute, right, where it was enriched in the primary mesangial cells, but not in the neighboring cells. That, to us, suggests that we're on the right track. Now, this is very early on. This is a pilot data. We're going to do far more work on this through doing the single gene knockouts and validations and inclusion of additional synthesizers. But I think even those two initial data sets should give you confidence that there is more to what's been reported for what might likely is going on to IgA. And I can rest assure you, neither one of those genes that I'm discussing here has been mentioned in the field at all. So I'm looking forward to the next stages as we validate those results to see how LeaPFroG was able to make progress on this problem that has been plaguing the field for some time now. This is where the words of Dr. Peter Slavin for my days as a junior faculty at Harvard and MGH come to mind, because he made a very when we were doing orientation, he gave a very key advice, which was that, get comfortable walking into the rooms where you don't know anybody's names and nobody knows your name. And if you work hard and stay true to what you're actually trying to do, eventually you'll know everybody's name, but nobody will really know your name still. And your goal is that before you leave MGH is that you start walking into the rooms where everybody knows your name, but you don't know anybody's name. And then the cycle starts again, right? So with that sort of two feedbacks in the back, I was fortunate that through the NID KK support to Susan, for the U54 grant, I was able to attend ASN, where, the first thing I noticed was just the thousands of new people, right? You walk into this hall, which is shown on the left. I don't know any one of these people, perhaps a handful of the people from Northwestern, but that's okay. I'm used to those kind of environments, right? So then I was fortunate to be able to interact with through different sessions, see what is the field doing on IgAN? It was where is the sort of current state of the art technologies on? And one of the things that I learned from this particular ASN was that there has been a lot of progress made on IgAN. But one thing that did stand out to me was that I think we're still missing a few things, because kind of things that we're identifying, for example, say whether it's the BAFs or the other sort of upstream markers, when you look at the actual data, when you really dig into the data, it's clear that those are, if proteinuria is the marker for what's going on in these patients, it's clear that they're not really capturing perhaps what's really truly going on. Because as you can see on one of the images that I captured on my cell phone, any of these trials that you look at, sort of next line of therapeutic, it's clear that in the initial stages, the therapeutic is having a great effect on lowering the proteinuria. And as long as the patient is kept on those drugs, the proteinuria levels are manageable and they actually are maintained at steady state. But as soon as you remove that proteinuria, the levels, so as soon as you remove the therapeutics, the proteinuria levels just shoot right back up. So it's not like these drugs are really curing the IgA, they're helping you manage it. So, I haven't done the statistics yet because there's not enough data points, but I think to me, at least looking at the slope from when it's actually lowering versus when it's going back up, we're probably able to make a case that, in fact, the underlying disease state may actually be worsening, because your rate of, when you stop the drug, how fast the proteinuria levels rise should indicate that it's probably still undergoing, continuously progressing worse and worse. So that to me suggests that we need an alternative, we need additional approaches to start looking from a different angle, right? These are great for now for patients who need anything to be able to make progress in IgAN, but there's need for more. And for that, we need to start asking the right questions and we start deploying the right technologies. And with that confidence, the other thing I wanted to be able to say, okay, where else can I help, right? So let's take that critical feedback saying, do nephrologists have existing problems that can utilize this kind of a technology? And one of the things I came across after going through many of the different posters across the 3 different days was that there is a lack of podocyte enrichment, right? So that's a great challenge in the field. Well, for example, it has to do with the biology of these cells. So for example, podocytes are terminally differentiated cells, as are many of the kidney cells with limited capacity to proliferate. So with limited access in vivo, because they're located deep in the tissue of the kidney, it makes it hard to be able to do any sort of imaging or manipulation in live animals. And the other problem is that if you do try to take podocytes and try to culture them ex vivo, they quickly de-differentiate in vitro. And the field knows that they'll take on this, what's called the cobblestone morphology, right? It's no longer resembling what the cellular state was in the patient itself. So that de-differentiation can make it difficult to culture podocytes. And so that suggested to me that we need rapid and efficient methods for podocyte enrichment from animal and human kidneys are needed. And it's not that there doesn't exist existing approaches that can do this. There's the differential sieving or microbeads or immunoabsorbations, but these are not really actually optimized. And as you can tell, if you really quantitatively look at what are the sort of enrichment in terms of yield or purity or cell health, these are not really great approaches to be able to make significant progress in trying to understand the podocyte biology and then link it back to the pathobiology of a particular disease of interest. So there's room for more. Now, one of the sort of always the pushback is, well, why couldn't we just use FACS, right? Well, that's true. You can use FACS, absolutely. You can use FACS for doing this, so this very problem. You can use FACS to functionalize the podocytes and then do the sorting and enrichment. The challenge, of course, is that how much access do you really have to the FACS facility, right? So it's not that, for example, Northwestern doesn't have the funds. I mean, we were one of the top ranked university with last year have over $1.05 billion of research funds that were secured. And that allows us to build numerous, top of the state of the world facilities, including flow cytometry facilities with capabilities that are probably a dream of many of the people, right? But unfortunately, with high sort of capacity and capability also comes the high demand, which means for average person who just wants to have a look at what's going on with this particular small problem, you're going to be in the queue where there are people ahead of you with more resources who are going to just be ranked higher, right? And the other problem is that you may not have that flexibility to be able to just do it when you actually want to be able to do it. So our approach is that let's build tools that democratize that kind of capabilities and make them readily available as the student wants to do, right? If I have an idea, I want to be able to start processing and generating preliminary data to see, was I wrong? Where can I improve? So we can actually cut down on this iterative cycle of testing and validating and reevaluating. And for this, I want to introduce LeaPFroG NephroSort, as I mentioned at the beginning. And this is a design that Daniel Wang has come up with, which is, he's a senior, sort of act like a CTO. Right now, he's a group leader at Chicago BioHub, and we're grateful for his support to be able to build these devices. And one of the things that we wanted to be able to demonstrate with this device is that it will have a broad spectrum analysis capability so that, once the device is ready, it should have a plug and play format and it's easily scalable because we can utilize injection molded based production to be able to produce as many devices as needed, as much as the community needs. And one of the key features of this device is no longer just a sort of sitting on a schematic in a CAD, right? We've actually produced this device already, and I wanted to be able to compare, to show the size comparison of this device, right? As you can see, these are my 6- and 8-year-old sons, and in their hands, you can see what these devices look, right? Both the top and the bottom part of these devices, and sort of symbolizes that I want to be able to provide these devices to parts of the world where perhaps the resources are not as readily available. So the limitation is not the availability of the devices. All you need is a curiosity and a question in mind, and you can be able to pursue these kind of analyses without being limited by resources. And of course, we're planning a feasibility study to make sure that we can demonstrate that the capability of NephroSort is on par, if not better, than the existing technologies. And this is one of the sort of planned study that we're doing, starting with, the first initial one is just to be able to show, can we, take a mouse model and take its kidney, make single cell suspensions, and do the particular functionalize it with any antibody that we want, and hopefully have to do with nephrin, and then be able to show that can we enrich a population of podocytes and what is the purity, and then use a whole bunch of metrics to be able to comparison between the existing FACS approaches versus the NephroSort approaches. And that data should be coming in shortly. So as you know, we're generating that data, one of the things I want to leave with near the end of this talk is a challenge for the field, is that, you guys are kind of being left behind. If you look at the human atlas, right? Right now, you're not on par with where the rest of the field is across other tissue types, right? So, and this has to do with, if you look at the total number of single cells that have been analyzed for kidney, right, in the human atlas, it's about 0.6 million. That's far less than even the pancreas, right? Or I mean, we're not even nowhere near where the heart is or some of the other organs, right? So that means that we need to, let's utilize the capability of these additional platforms and technologies to be able to enhance the data sets, so we can utilize those data sets to be able to come up with better hypotheses, to be able to address more challenging questions that relate to kidney disease research. And lastly, I want to kind of share the key component of LeaPFroG is not just the fact, we can generate these devices and the capabilities. It's also the fact that LeaPFroG brings in people with different expertise into the same room and then start asking questions. What are the existing tools that are already readily available that we can deploy? What are the limitations of those tools? And how can we, as a community, then come together to be able to build better tools? And when it comes to cracking hard problems, that kind of intangible ability to be able to put expertise in the same room never shows up on any standardized sort of bar charts based early evaluation metrics of new technology. So I think that always goes missing when you're trying to evaluate whether we want to invest in these kind of technologies going forward. And I just wanted to be able to highlight, that will never show up on any sort of bar chart that what LeaPFroG can do unless you have the right people asking the right questions. And with that, I want to end by thanking everybody who's on the call for your time. And I especially want to thank Professor Kelly and Professor Quagan, because, in my experience, when you have new ideas, you typically come across two types of people, right? Those are the ones that will look at, your idea and the existing state, and they'll decide, does that make sense, right? And then they'll decide whether it's good or not. But then there are rare few stars who look at the ideas not for what they are right now, but what they can be. And that's the group of people that you want to surround yourself. And for that, I'm so grateful that I not only have one, but two believers and champions who see the technology and its capabilities for not for what it is today, but what it'll be for tomorrow, because that kind of vision is necessary for patients all across the world who do need better treatment options because their very lives depend on that. So with that, I would like to thank the funding agencies, our sort of collaborators on the resource core. And I also want to give a big shout out to two individuals who have been super helpful with organizing this talk, particularly, Esme, who's worked tirelessly for the last 3 months to make sure, everything was running smoothly. And the second is, which is a critical component for this kind of disease research, is the patient advocacy. And for that, I'm grateful for Arnold Davis. He's been on every single call that we've had as a group to be able to, sort of weigh in, okay, what is the patient perspective? How can I help? You will never find, in my opinion, a better advocate and a better cheerleader than Arnold Davis when it comes to kidney disease research. With that, I'll stop and thank you all for joining again, and I'm happy to take any questions.
Wow. So first of all, thank you, Randy. That was a true tour de force and really a very fantastic presentation, although you said far too many nice things about me, so we have to remove those from the recorded version. We are going to open it up for questions. I just want to say, from my perspective, not being the technology expert, I'm just holding one of the LeaPFroG, so it is truly an amazing device. And as Randy has pointed out, I think today, I think the applications of these technologies are really endless, and not only for difficult clinical questions, so the IgA absolutely is a difficult, complex problem to solve. However, isolating rare cell populations and, we predict that their transcriptional and their epigenetic signatures are going to be better preserved because of the ease, the speed, and the ability to really batch these. So we do have a question, and people can either ask to, raise their hand and we'll unmute you, or you can ask it in the chat. I've got one from Dr. Dalal, who asks, can intracellular targets be used to sort cells via nephrosort, or does it have to be a cell surface target?
No, absolutely not. So the only limitation is what's your end use of that population, right? So if you want to do some downstream where you want to have, for example, if you want to do single cell sequencing, then you probably want to maintain a live cell population. So you're limited with using a cell surface. But if you don't care for live cell populations, you can work with dead cells afterwards. Then we can do intercellular. Because part of the problem with introducing, now that's only a current problem, by the way. I should mention that gave us about a year because we're working on some other technologies in parallel that will hopefully allow you to be able to introduce your detection reagents into live cells, intercellular as well. For currently, we're limited because we have to fix and permeabilize the cells to be able to introduce the immunomagnetic nanoparticles that form the basis for cell sorting.
Okay, we've got another one from Gary Gerlach. Do you want to unmute yourself, or would you? It's an excellent and interesting comment and question. I can read it out. Says it might be argued that FACS analysis of podocytes will change junctional structure, the slit diaphragm. Fluid flow, shear stress is essential to podocyte function and architecture. Many labs have attempted FACS of podocytes without fluid flow. For instance, during FACS analysis and sorting, gene expression will change in podocytes and junctional protein will likely be lost from slit diaphragms. I imagine there are different subpopulations of podocytes, but how to identify them still remains elusive. I wish your system the best in attempting these endeavors, but the system might be adapted to include fluid shear stress. Oh, sorry, Gary, I just saw red. But maybe Randy could talk about the possibility of including flow, shear stress into the LeaPFroG technology.
Yeah, this is where really the feedback from the expert from the field is really, really super valuable. So thank you for sharing that feedback, because that makes me understand, okay, what is really the key sort of thing that's missing in the way we're looking. So we can definitely build that. So for example, one thing that we have known when just doing comparison in the previous project with FACS versus our LeaPFrog version of cell sorting is that because of the time frame requires, often cells will have changes in their homeostatic state that is not due to the disease state, but just for the fact that they were just sitting on ice or sitting in a buffer for cell sorting long enough that actually changed their homeostatic state, right? So if we really want to capture what are the differences in expression differences relating to a disease state that is actually properly recapitulate what's going on in vivo, we need to cut down on these extraneous sources of variability. So that's where, the more information you can share with us, the better refinement we can introduce to our tools to be able to capture, to ensure that we're capturing the population that is reflective of what's going on in vivo.
Yeah, I'm going to ask another question, but Esme, I might have you unmute Gary in case he has another comment he wants to make after Randy.
I'm unmuted now, I think.
Oh, you are? Okay. Do you want to? Yeah, any comments or follow-up?
I just think podocytes are one of the trickiest cells to try to FACS because their function is relying on the architecture, and as soon as you break that junctional component, the podocyte's going to go into a stress response, and it's a really tricky paradigm to work with on that cell type FACS.
Yeah, agreed. Yeah. So we are going to compare head to head with looking at gene expression, not though, of course, at architecture per se, but it might give us some clues as to what the differences are, including the number of cells that are isolated per kidney. Yeah. No, it's a great question. Okay, Javid, I see your hand up. Do you want to unmute? Oh, you're unmuted, I think.
Yes, good afternoon. Can you hear me?
Yes.
Yes, perfect. Yeah, good afternoon, everyone, and thank you for the nice presentation. Can human antibodies from serum be labeled with the nanoparticles and then use those to isolate mononuclear cells?
Yes, absolutely. Yeah. So if you have an antibody, you understand these, or if you know the species, that's all we need to know. We can actually then have coupling reagents that actually tailor to that particular species, for example, or that type of antibody to ensure that we can load the magnetic nanoparticle to that specific antibody and its specificity.
I see, but the bulk human serum with antibodies, can that be labeled and then used to isolate?
So bulk serum, yes. I mean, I think maybe perhaps I'm missing a little bit more details on what specific applications you have in mind. I'll be more than happy to discuss in details offline, and then we can I'm certain that we'll figure out a way to whatever the problems may be in that particular situation.
Yeah, sure. So
Yeah.
I will reach out to you.
Yeah,
you.
yeah. I
Thank
guess
you.
I'm just going to follow up on Javid's question there because I think, yeah, it would be like isolating. So actually not knowing what the antibody is
Correct. Yeah.
necessarily, right?
Yeah,
From
exactly.
a human, but knowing that there is something circulating in the blood that we think is
Want to
causing
isolate.
the problem, and we want to identify. So could you label multiple, presumably multiple unknown antibodies?
Antibodies.
Yeah,
But a
just
known cell population,
yeah.
presumably.
So one of the
The
actually now
reverse.
that additional information I should mention that one of the assays that we've developed, so completely separate from this project that discussed, is that in collaboration with Amgen, we were building systems to be able to capture antibodies on the cell surface secreting cells, right? So if monocytes are secreting these antibodies, we can actually capture those different monocyte population as they're secreting those antibodies and enrich those based on the level of a particular type of antibodies that you're looking at, right? And then actually then characterize those monocytes as to what is in their particular makeup that was actually making them secrete this type of antibody or this combination of antibodies.
Yeah. That's great.
Cool.
So yeah,
yeah.
I think
But it
we
looks
should
like, yeah.
we should look into this a little bit more.
Yeah, I think that could be really interesting. Yeah, there could be lots of applications if you were able to do that where we think. Yeah, and maybe I'll follow up. I know we're getting close to the top of the hour, but I was really interested, and I'll have to read the paper on the secretory piece of it. So what are you actually sorting by for the secreted?
So the way the secrete assay works, which is upstream of LeaPFroG, which is that we actually functionalize the cells to be able to capture as the antibody is leaving the cell. So it'll be captured on each individual cells, and depending on the different secretion rates, what ends up happening is that it labels on the cell surface different levels of the antibody that was being secreted, right? So now we can then capture with an anti, for example, in this case, we were interested in interferon gamma secretion. So then we use anti-interferon gamma antibody to then say, okay, what are the cells that were high secretors of interferon gamma at single cell resolution, mind you, and then just have characterization of those at a bulk level to be able to say, okay, what were the gene changes and underlying perturbations that led to the high or low levels of interferon gamma secretion.
Yeah. Okay, cool. Okay, we have another question from Penelope. And yeah, we've got two. Do you want to unmute, or maybe I'll just read them for sake of time? So the first question, could you comment more on exosome characterization compared to the standard methods?
We just published a paper. It's in Nature Nature Communications. This came out earlier this year. We can do a comparison with the existing methods, and we showed that in terms of purity and enrichment, and then just be able to demonstrate the usability of those exosomes. We were the LeaPFroG version or the ExoPrism version of the devices outperformed the existing methods for enrichment of exosomes. I'm happy to actually. I'll forward that paper, Penelope, for your review as well.
Great. And now we've got another question, which is interesting, from Dr. Bradford. Will these applications be adapted for spatial analysis in intact tissues?
That's a fantastic question. In fact, in the context of IgA, even, right, we were even thinking, even in the podocytes, as you were mentioning, right, Gary made a good point that, we may not be able to do anything with those cells, once they are actually out of it, but we can still have the spatial information as they were when we actually made the single cell suspensions, right? And there might be a way for us to actually be able to leverage that, to be able to build data sets that capture the diversity of states that exist in these population of cells.
And would you be able to, I guess, magnetize and look at levels of expression in situ?
Yes.
Yeah.
So in fact, in my previous life, I had done that already, and for where we did some screening, and we were able to actually go back to that, where we did a whole panel of phenotypic analysis, and then we had exact location, where was that cell that had this particular features. We can always go back and then extract the genomic DNA or RNA, whatever needed to be able to then do the characterization of that particular cell. Yes, those capabilities can be built.
Terrific. Well, I think we are at the top of the hour. So, I would like to thank you, Randy, for a really fabulous presentation, really exciting. And I want to thank the audience for being fantastic, great questions, and I'd like to thank the OKRA team for organizing all of this. So thanks so much. Have a great afternoon, everyone.
Thank you for having me. It was great. My pleasure.
Bye. Bye.